Hello, welcome to Biology Videos at TDA. We're going to do 13.10 sliding filament theory. We're going to be looking at the actual process of the muscular contraction in a bit of detail, rather than just looking at the sarcomere itself and the myofibrils. First question is, it's a joke, how much does muscle contraction cost? I'll let you have a think about it. You will know the answer to this. It's a molecule. I'll come back to this at the end. Okay, so what we do in this lesson is we're looking at the model of muscular contraction, the role of ATP, and then the links between the mechanism and muscle control. So, before we go on, here's some questions. I want you to write these answers down. I don't expect you to get them all, but I'm going to review these at the end of this video, and then we're going to see how much you got. So pause the video, see if you can answer these questions now. We should be able to have a go at some of them. Okay, so we'll come back to them shortly. So, a reminder of muscular structure. We will be looking at all of this in this lesson, and we're looking at the overall depolarization from the synapse and how this goes down. Remember, we've got these T tubules. We're going to look at these. These are like these folds of the sarcolemma, which is the membrane of the muscle cell, the plasma membrane, which is the sarcolemma. They're these folds that then go all the way down, and you've got these myofibrils, which is just labelled here as fibrils, which are these bands of sarcomeres all joined up together, and this is all within the cell itself. Okay, so a reminder of one sarcomere. So this is a photomicrograph, and you can see the light and the dark bands. Sorry, the dark bands here, the light bands here, this is the Z line. And this is what it would look like under a microscope, and you have to be able to label and understand. And we've got this thick section here, the myosin section, and then the thin section, the actin, and that then forms these dark and light bands. And what you have to understand, it's a bit like if you take your hands out in front of you now, and then you slide your fingers into each other, and then out again. It's just like that. That's just how muscle contraction works. So it can only contract so far, and then it pulls out again. And it goes in and in and in until it's fully contracted, and then it can be pulled back out again. So, first question, let's see if you can answer these. State what happens to the lengths of the following when muscle contracts. What happens to the A band? Is that going to change length? Is the H zone going to change length? And is the I band going to change length? So just say, is it increase, decrease, or no change? Now you're going to answer this. So you're going to go to this link. It's on Show My Homework. You don't have to type it all out. If you go to Show My Homework, it's down the bottom. You can click it, you can watch it, and then you can mark yourself. It's quite a really nice animation, and it's also showing you this hired MH education site that's got loads of different animations. So if there's something you're struggling with, you can maybe bookmark this in your browser, and then you can come back to this for something. If you're ever struggling, you can go, do you know what, I'll try and see if there's a good animation. So you can mark yourself. Okay, so let's start looking at the process a little bit more. So we've got this synapse here. So this is the sarcolemma, so the outside of the muscle membrane. So just as we learned in the last few lessons, we're looking at action potential. So we've got an action potential that's arriving, and it's described down the bottom, and on the next slide it's all written out for you. So I'll try and describe it myself first. So you've got an action potential that arrives. That's going to cause this influx of calcium ions. That's then going to cause the vesicles to then move. It's going to cause exocytosis. This is going to spit out the acetylcholine, the neurotransmitter. That's then going to bind to receptors on the sarcolemma. So we know all this bit from the last lessons, but this is now going into the new bit. Okay, so acetylcholine has been released. That's going to then be broken down, and it's very important that's broken down for the same reason we looked at neurons. Is otherwise you're going to have overstimulation of the muscle. If you don't continuously break down the acetylcholine, you're going to overstimulate the muscle to contract over and over again. So you're going to have to break that down into the acetyl and the choline. That's then going to cause the generation. So it's going to bind to receptors on the sarcolemma. It's then going to cause depolarization. It's going to then cause an action potential to spread down the membrane itself. Now this is like a shortened version, it's not to scale, but this is going to then spread deep within the muscle fibre to all the different myofibrils. So all the myofibrils can contract at once. Now that's what we call a motor unit. So all the places in which this binds to is called a motor unit. So you can have contraction of one particular neuron that binds to this section, that binds to another section, and another section, and another section somewhere else. That's a motor unit, and you'll recruit a motor unit to do exercise. And the more exercise you do, and the harder the exercise becomes, the more of these you're going to then contract, so you're stimulating more muscle fibres at once. So the reasons how you can just lift something light up like a cup compared to a weight is you're only activating a few of your motor units. So a few of these synapses are going to occur, so you're only going to have a little bit of activation and then a little bit of a muscle contraction. You can regulate that. And the more fatigued you get, the more they get used and used and used. So you're going to keep recruiting more and more and more. So that's why exercise to build muscle and exercise, the more you do, the more fatigued you get, you're actually activating more motor units. And part of your improvement in exercise and fitness from doing exercise is because you have actually improved the motor control of this before you actually increase the strength of the muscle fibres themselves. Okay. Right, enough on that. Let's go back to this. So you've got the sarcolemma, the depolarization. That's then going to get to the sarcoplasmic reticulum. That's going to cause channels within the sarcoplasmic reticulum to open and diffusion of calcium ions that then is going to get to the muscle filament. So we're going to look at this. So on the next slide there's a full recap, but I'm going to recap it now. Action potential, neurotransmitter goes across. That then binds to receptors on the sarcolemma. Action potential spreads down the sarcolemma and then down the T-tubules. That gets to the sarcoplasmic reticulum. That causes calcium channels to open. Calcium diffuses out of the sarcoplasmic reticulum, which is essentially the muscle version of endoplasmic reticulum, into the sarcoplasm, which is the muscle version of cytoplasm. That's then going to start binding calcium ions, are going to start binding to the muscle fibres themselves. Okay, so we've got the basic idea of how do we stimulate the muscle contraction. We're now going to look in a bit of detail the muscle contraction itself. You can pause the video, kind of all I've put there is in this, so you can pause the video and read through it if you're a bit confused still. Okay, so what I want you to do as I do this is a guided visualization. So that means I'm going to talk through the stages. I want you to draw a symbol to represent what I am saying, and that's the idea. Is it's going to be like a memory jogger. It's quite a common technique used to kind of remember things. So for instance, when I mention calcium, you could draw a little bone like a femur. When I mention myofibril, there might be a way mayo, I don't know, mayo, a little pot of mayonnaise, whatever. So the more creative, stranger this is, the easier it is for you to understand. So as I go through each section, and I will bullet point everything I say at the end, draw a little image just to start with, so you can kind of keep up rather than having to scribble down of what I'm saying. And you can pause the video, think of something, draw it, etc. Okay? So, a couple of words. Again, this is for you, a bit to do with the glossary task. You can pause the video, and if you're a bit unsure of some of the terms I'm using, you can pause it and reread this. Okay, so here's my animation that I've got. I know you've seen some other versions, so I'm going to try and talk you through this one. This one goes quite quick. I haven't managed to slow it down, so I'll do my best. So, the start point, as we know, is going to be calcium. So if you look kind of in the top right hand corner, and then I'll just get my mouse cursor, so you should be able to see my mouse cursor now. If you can't, I apologise. In the top right hand corner, the calcium ion is then going to come down, and that's going to bind to troponin. So the first step is calcium is going to bind to troponin. Step 1. Next, troponin is going to change shape, and that's going to then pull on the blue fibre, this tropomyosin. That's going to pull on that. Now currently that's preventing this big red myosin head from binding to the actin and pulling on it and doing the muscle contraction. So that's covered over at all times by the troponin and the tropomyosin. So if we bind the calcium to the troponin, that's going to pull on the tropomyosin and expose the binding site. So step 1, calcium binds to troponin. Step 2, that then changes shape, pulls the tropomyosin away and exposes the binding site of the myosin on the actin. Okay? That then allows the myosin head to form a cross bridge. So the point at which it binds onto the actin, we call that a cross bridge, where that binds onto it. and that cross bridge is then going to be pulled down. The shape of the head is going to be pulling a bit like your two fingers interlocking, and the whole sarcomere is going to start to shorten. All those the distance between the two Z lines is going to get closer, and the H zone is going to start to disappear. Then we're going to have to break that cross bridge, and energy is required to break that cross bridge. So ATP, which needs to bind to the myosin head, is then going to be broken down by ATPase. That's then going to cause the breaking and that, like the re-preparation again of the myosin head to do another muscle contraction. So energy is required to break the cross bridge itself. Now, if you haven't got the joke here, ATP, how much does the muscle contract? ATP, 80p, 80 pence. Anyway, that was the joke. So you need ATP to break this cross bridge that can then prepare it for the next muscle contraction. This can happen 100 times a second. It's really, really very fast. Okay, and I'll bullet point these steps and go through again this in a second. Also, I've got in my animation, if you have a look, magnesium. Magnesium is used in muscle contraction to regulate calcium, to prevent the overstimulation from the calcium ions. So we don't really go into this in OCR, so don't panic about it too much. It's just on this animation that I've managed to find. Okay, so let's look at the steps. So the first step is action potential gets the motor plate, release acetylcholine, blah blah blah. We get to the point in which calcium is ready. Okay, so you kind of knew step one. Calcium binds to the troponin. That causes the tropomyosin to change shape and be pulled away from the actin binding site. Tropomyosin moves away. We now form a cross bridge where the myosin head binds to the actin. That then pulls the actin filament along. That's your kind of contraction. That's your actual pulling, the movement bit. And then energy from ATP is used to break that cross bridge using the ATP enzyme in the myosin head. It's now been reset, and then we'll repeat this process over and over again. Okay? There's lots of animations. If you're still struggling with this, there's lots of animations you can use if you haven't understood my understanding. But this is a formation. Hopefully you have a little picture to represent this, and that should make it easy for you to understand. What I suggest you do now is make sure you have a picture for all of these written down on your page, just a little picture. Now have a go at saying out loud, pause the video, say out loud all the steps, see if you can do it off by heart. You need to be able to understand this whole section off by heart, I'm afraid. Okay, so an activity you can do, now that you've kind of got that, is try and put these in the correct order. So pause the video, have a go, just literally list the numbers from top to bottom in the correct order. Okay, so if we go through this now, so we've got the action potential arrives at the motor end plate, which releases acetylcholine. That's going to bind to the ligand-gated receptors. That causes depolarization. That's going to then spread through the T-tubules to the sarcoplasmic reticulum, which releases calcium ions. Calcium ions are going to bind to troponin molecules, changes the shape, moves the tropomyosin away from the actin binding sites. That's going to then make the actin myosin cross bridge to form. Really important word that we use, cross bridge. That's a key word in this. The myosin head can now attach to the actin filament. Now we've got the power stroke. So we've got the binding site, the cross bridge, and the power stroke. So the power stroke is the movement. That's going to bend backwards, pulling the filament along. That's going to then attach and detach from the actin, causes the filaments to slide on another. So that attach and then detaching, that continuousness is going to slide that along. And that's going to decrease the length of the sarcomere length. But the actual actin and the myosin, apart from the myosin head kind of changing angle, there's not actually just a change in length of the fibres, the filaments themselves. It's just how they're slid over each other. And then number 7, the cross bridge is going to be broken using the hydrolysis of ATP to ADP and the phosphate group. Okay, that's mostly it. So if you can get that, you should be pretty happy. So, or ignore that, sorry. So let's go through some kind of interesting facts that we looked at the beginning. So why is the duck meat dark and chicken meat white? Or more red, darker, and the meat and chicken red? So these were the questions asked at the beginning. So if you've eaten either of these, if you're not a vegetarian and you've eaten either of these, if you were to eat both of these plain, which one would probably taste nicer? Again, it's opinion. But quite a few people would go with the duck, the duck tends to taste a bit nicer, and that's because it's red because it has more fat stored in it. Chicken is a very lean meat. There's not very much fat in there, which is why it's white. Rigor mortis as well. What's this rigor mortis, if you haven't seen this before? So when animals die, including humans, when animals die with muscles, they go stiff for a very short period of time. Now rigor mortis is where your muscles contract, and then you've died, and then your muscles go stiff. Now later on you kind of go a bit softer. Now have a think about why after death might your muscles go tight. Have a think. So the reason is because if we go back to our sliding filament theory, if I just skip back to this, to my image, energy is required to break that cross bridge. So after we die, we stop making ATP. We stop doing aerobic respiration because we're breathing. Any organism that is not doing aerobic respiration will then not produce ATP. So therefore, the myosin head stays bound to the actin filament. Hence, the stiffness. So if you were to pull on the arms and legs of a dead animal or a dead person, you'd have to pull them apart. And you could do it, but they would be stiff. So that's why, because it's still bound to it. So force is required to break that apart. Now, eventually, later on after death, then the proteins themselves start to break down because they're not being maintained. So therefore, rigor mortis only lasts for a little while. Okay, so fast twitch and slow twitch. What's the difference? A reminder, so that fast twitch is going to be more powerful, it's going to fatigue more quickly, and slow twitch is going to be more resistant to fatigue. And if we were to look at what they look like under the microscope, just kind of an image, slow twitch fibres are going to look more red because they're going to have more myoglobin stored within them. So here's the answers to all of them, and I added a couple more in there. So there's more fat in duck breasts, in duck meat. What is depolarization? Well, that's the change from being polarized in the electrical activity. What enzyme releases energy from ATP? Well, it's ATPase found in the myosin head. How do myosin actin allow you to move? Well, the myosin head forms a cross bridge, pulling on the actin in a power stroke, and that shortens the sarcomere. What does tropomyosin do? Well, it covers the binding site for the cross bridge. Why is it important? Well, it allows us to then reset the myosin head and then do the next power stroke. If it's never, if it's always open, the head will always bind to it, so you'll always have this kind of rigor mortis stuck. In muscles, what are the IH and A zones and bands? So the I band is the actin, the length of the actin. The H zone is the space between the actin. You can just check a sarcomere to get this. And the A band is the length of the myosin. So both the I and the A aren't going to change, but the H zone could do as the muscle contracts. Fast and slow twitch muscle fibres, so fast fatigue fast but more powerful, and slow fatigue slower but are less powerful. Okay, that ends it. I'm not putting any questions in today because there's quite a few activities here. I'm going to just touch up for the next lesson. You can have a nice break now. I'm going to touch up on sensors. I know there's a little bit more in the textbook, so I'll try and explain that a little bit more. And I'll kind of give you a bit more of a background information about different things here to do with muscle fibres. So that's it. Thank you very much. So if you just submit what you've done the normal way, and have a wonderful week off. If you're slightly behind, can you try to catch up as much as possible? We're going to set you a kind of a very non-stressful chapter test on this when we come back. So once you've done this lesson, you can get this done today, fantastic, or over the weekend. You'll have nothing to do next week. Thanks guys, stay safe.